So every Thursday with OceanWise, we're excited to be featuring the great work of OceanWise research and finding out more about what's going on at this time. And so today we are joined by Dr. Adam Warner to share his insights on ocean CSI, tracking a killer whale. And we're really excited to hear more. So as you're just getting signed in today, for all of our audience members, again, we have the chat open for you. We'd love for you to just select to host and presenter, then both myself, Danica, your host from online learning, and Dr. Adam Warner will be able to see all of your comments and questions from today's program. If you do have a specific question, we would love for you to open up the Q&A just at the bottom of your chat window. And you can put your questions in there. That's a great way for us to keep track of what those questions are and make sure that we answer as many as possible for you today. All right, so I think it's just about time to get started here today. It's 1 o'clock on my watch. And so to start off our program today, I would first like to say how grateful I am to have the opportunity to teach, learn, and live on the traditional territory of the Squamish, Musqueam, and Tsleil-Waututh people. Since time immemorial, all the way to now, they have been incorporating storytelling and observation and science in all of the great work that they do. And we are so appreciative to be able to learn from them and incorporate those practices in what we do for OceanWise education and OceanWise research. So thank you to our audience for spending your afternoon with us and learning more about OceanWise research and the work that we have going on. And without further ado, I will turn it over to Dr. Adam Warner, who has some great stories to tell with you today. I'll be on mute. I'll be monitoring the chat, helping pass those questions throughout the program, and I'm really looking forward to learning more.
Great, well, thank you very much. So yeah, I'll just say hi to everybody, and thanks for joining us for this OceanWise event. And I'll just try to share my screen here in a second. And we can talk a little bit about DNA and how we use it in the conservation world to help our conservation research. And I think this is working. Okay, so yeah, so I'll be talking to you a little bit about how we use DNA to track killer whale populations. I'll talk a little bit about DNA itself and really kind of give you a bit of an intro to that. Hopefully it doesn't get too lecture-y. I'll try to keep it a bit light. And then and also talk about some of the other work that we're doing in the Marine Mammal Research Laboratory. So this is a bit of a busy slide, and that's because we are very busy doing a lot of different things in the research department. So we have the BC Cetacean Sightings Network, which promotes citizen science. We have the Whale Report Alert System, which helps identify and alert mariners to whales in the area. On the North Coast, the Cetacean Research Institute initiative promotes the BCCSN, but also does some harbor porpoise habitat use, wintering humpback whale study, and the North Coast Humpback Whale Catalog. We do a lot of field work. You can see there closer to the right. So looking at the health and condition of killer whales and humpbacks, looking at pinniped diet and impacts of underwater noise as well. In the Arctic, looking at impact of underwater beluga communication and behavior. And we have the Wild Killer Whale Adoption Program, which raises funds to support a lot of this research on wild killer whales, which brings me to the Conservation Genetics Lab, which gets a lot of help from the Wild Killer Whale Adoption Program. And what we're mostly doing is looking at genetic variation in marine mammal populations, looking at killer whale mating systems, identifying stranded carcasses, looking at killer whale diet. We might be doing a bit of that in the future, and using DNA to kind of link a lot of a bunch of the other research that we're doing together. And I'll get into how we do that in a little bit. So BC is home to three distinct types of killer whales. So there are, and you probably heard this from some other talks if you've been following along this series, but we have resident killer whales, which eat fish exclusively. And we have two populations in BC, the southern resident population and the northern resident population. There's the Biggs killer whales, which you may have heard of as called transients as well. And they eat marine mammals, and seals are their preferred prey. And these are a lot of times the killer whales that we see right around Vancouver, right up by shore, because seals do try to get away by getting on shore. They hang out on shore or near the shore, and so the killer whales come right in, and that's what they're doing. They're hunting. We also have offshore killer whales, which eat sharks, and that's one population from Alaska to California. And we don't see those very often, but they are of interest to us as well for conservation. And so for the southern resident killer whales, they have a pretty wide range. In green there, you can see on the far left, they range all the way from Haida Gwaii down into Washington, but they have a really critical habitat right around the southern BC coast and into Washington. The northern residents range further north, even into the waters off Alaska, and they don't really go as far south into Washington. So the two resident populations don't overlap by too much, but they do for, at certain times of the year. And then we have the transient or Biggs range, and they just kind of go everywhere. So you'll see them up around Alaska and then down in California as well. And depending on the killer whales themselves, some do stay more localized, but definitely some range quite widely. And the status of these killer whale populations is one of the reasons that we're doing research to help with their conservation efforts. The resident population is about 380, and 300 or so of those are northern residents, and they're threatened. That's still a low number. And the southern residents, which we hear a lot about, I believe they're 72 right now. So they're actually endangered and critically endangered. The Biggs transient killer whales, they're at about 500 in population size, but that's still not a lot of killer whales, and so they are threatened. And then the offshore population is threatened at around 300 individuals as well. So how can we aid conservation efforts through genetics? One way we can do that is understanding more about how genetic diversity, which can help fight off diseases and infections and all sorts of other things biologically, how that is transferred through the population. So paternity is one of the ways that we can look at that. We can identify fathers to determine how extensively males in the population are mating, which is important for genetic diversity. And I'll leave it at that right now because I'm going to get into that much more in depth later. But that's one way that we can really learn a lot. Also, we can identify killer whales based on poop. And so we have a stress hormone and body condition study ongoing. And what you can do is measure stress hormones in killer whale poop. And we can measure that between individuals. We can measure it from year to year. But one problem is if you find killer whale poop in the ocean, you also need to know which killer whale that poop's from if you want to link it to other studies. And we can use DNA for that. So Biggs killer whale paternity, which I'm going to get into a bit more in depth now. We really want to know who is the father because males mate and then they just go back to living with their mother and immediate family after mating. And so generally we don't see the mating events and we never really know who the father is. Mothers, generally we always know, but the fathers not really at all. And there's a big study in 2018 that looked at this in the southern resident killer whales. And since 1990, just two males sired over 50% of all calves during that time period. So they were, very active in mating, and now both of them have actually passed away. So we're looking at seeing what happens now with other males maybe taking over those roles, and just seeing what happens. But it's really interesting that just those two did so much of the mating, and so we want to see if that's happening in the Biggs population, which ranges mostly off of the BC coast. So using DNA samples, we can get a genetic fingerprint for each killer whale. And generally this is from a little piece of skin that we obtain using a biopsy dart. But we can also get that from killer whale poop as well. And that genetic fingerprint can be used to confirm maternity and also infer paternity. Oops. And then the other main study, getting into a little bit more, is the drone photogrammetry that the Marine Mammal Research Group is carrying out. And this involves flying a drone over killer whales in the field, and we can actually see interactions between the killer whales, foraging behavior, pregnancies, but also health and body condition. So we can see if an individual is healthy, how they're growing from year to year, even throughout the season. If they're observed more than once, we can see how the feeding during those sort of heavy salmon summers or low, if that affects their body condition. And during that same time, we have another crew led by Caitlin Yale, who collects poop samples and does this the stress hormone study that I mentioned before. So the poop samples are collected, stress hormone levels are measured, and actually these poop samples are sent off to a bunch of other labs too to look and see what the killer whales are eating exactly. We can even determine which salmon runs the salmon are coming from. Even see what bacteria makeup is in the gut of the killer whales. So you might have heard the term microbiome. For those of you who are a little bit older, it's a new sort of area of study. Kids, I don't expect you to know that, but maybe you do. But what we can do is link all that data together. But one key thing that we need to know is which killer whale it's from, because we can get all that information, but if we can't link it to sort of the observational data that we have, it's not quite as useful. So here on the right, we have a small sample of killer whale poops. It's just a little Q-tip swab. And at the time, I just knew this was a 2018 collected sample, southern resident killer whale number 5, and it turned out to be J38 Cookie. And this killer whale in the observational notes, it said that this J38 was in the area, so this one was good to confirm. But a lot of times, they're traveling in a pod, so there are multiple killer whales around, and that's where we really need to make sure that we're getting the full set of data that we can make that determination. So stepping back and just looking at DNA and seeing how we can actually use that to identify individuals, it's really about finding a pattern. And to find that pattern, I want to just tell you a little bit about how DNA, RNA, and protein are related. So in our bodies, we have DNA, which is a molecule you may have heard of that is a whole bunch of different letters, and they're all in a big long string. And you can see that on the top there. And not all those letters actually code for genes, but genes are basically blocks of DNA, blocks of letters that get turned into RNA, which you see in the middle there, by proteins. They make this RNA. So that codes letters that code for proteins that we want to make. And so then those letters in RNA are then translated into protein. And that's what helps build our cells and do everything that our body needs to do. But all that information is stored in that DNA. And that DNA, on the right there, you see that sort of double twisted, double helix, 2 strings of DNA. It's all packed into something called a chromosome. So that's sort of the whole big long string of DNA. And humans have 23 pairs. Those are what make up, you have 23 pairs in every single cell. And what's important to get out of that is we get, and killer whales get, 1 chromosome from our mother and 1 from our father. And we all know that not everybody's DNA is exactly the same. So 99.9% is all the same throughout, but we do have differences throughout, and that's what makes us all a little bit unique. And these differences can be, a bunch of different letters or bases missing or gained, but usually a lot of them are just one change in a letter. And these changes tend to happen at the exact same spot in different people or different killer whales. So there are these spots that we can look at and see what the letter is, and we get kind of a pattern. So at site 1, we get a certain letter. At site 2, we get another letter, and that pattern can help identify an individual. So here we have that sort of single spot on the chromosome that can be different is labeled with a different color, and you can see that the father has two choices in the sperm for what the father can give to the child, and then same with the mother. And so we can get these different arrangements or different hereditary patterns, or how the DNA is passed on from parents to kids. And these can cause changes that we see or don't see. So a classic example of one of these single nucleotide polymorphisms, which I'm just going to call a SNP from now on, they lots of times they don't actually do anything. So they can be outside of a gene. They can just do something that doesn't cause a change that we see. But one classic example is brown eyes versus blue eyes. And there's one spot in our entire genome. So the genome is all the DNA in our whole body. And there's one spot where if you change it to a G, it doesn't actually do anything if it just happens once. So you still have brown eyes. But if it happens twice, or you have two copies of that DNA where it's GG, you just have blue eyes. And it's as simple as that. And so sometimes, for that's something that we can just observe and see, okay, at position 3,532,000, whatever the exact spot is, that's a GG. But generally, we can't tell that. So we actually have to sequence the DNA, or basically get a readout of what the letters are at that position. So the way we do that is we take killer whale poop and we use chemicals to get the DNA out, and then end up just with pure isolation of DNA in a tube. And then what we can do is something called polymerase chain reaction. And I won't get into it too much, but it basically just takes DNA and makes lots and lots of copies of the stretch of DNA that you want. And you can visualize the DNA pieces on the right there on something called a DNA gel. And that shows you the different sizes, because they'll move a different length through the gel, depending on if they're shorter or longer. But that still doesn't give us the actual sequence itself. And this is just showing a PCR again. So you probably heard PCR being used for coronavirus, because it's making lots and lots of copies of coronavirus that they can then sequence, or do other types of analysis on. But we can do the same thing for making lots of copies of these pieces of killer whale DNA that we want to look at. And then when we have tons and tons of copies on the right there, what we do is sequence it. And sequencing has changed a lot, just even since I was in university. I wasn't in university in the 70s. I'm not that old, but but in the 70s, Sanger sequencing was sort of that was the invention of DNA sequencing, and the equipment was in the hundreds of thousands of dollars range. So pretty expensive, but the cost to sequence, say, the human genome was billions of dollars. So very, very expensive, and it took years and years to do. Then around 2000, there's a new technology that came out. And again, the sequencer is huge. You need rooms filled with these. The equipment was super expensive, so even now these are millions of dollars each. But you can sequence a genome now you can sequence all the DNA in a human body or killer whale for about $2,000. And it only takes weeks, so that's pretty cool. But you need the $1 million or a couple $1 million to actually have these machines. But there's been sort of another big jump in technology, and that's been something called nanopore sequencing, which is a little bit more in your average lab's budget, where the equipment is just $1,000. And you can still do the cost per genome of about $2,000, and it's just weeks of time still. And I won't get into this slide too much, other than nanopore sequencing it's sort of almost science fiction. It seems like something you'd see in Star Trek and think, oh yeah, there's no way that could ever happen. But we can actually do this. So the way it works is DNA is pulled through a little tiny nanopore, which is basically a little protein. And as the DNA goes through, it's split into each separate strand. And as the DNA goes through, it just changes the electric current a little bit that's running through there, and that's measured. And you can see there where it says AC, TG, HG, CT, just to the right of that blue pore. A detector actually detects that change in current and gives you this readout of what the bases were that went through. And so here, working from home, I actually have the sequencer at home. It's meant for the field, but in this case, the field is part of our bedroom. And so I have the sequencer here, and you can see it running. Hopefully the video is working okay, but I don't know what those lights actually mean, but it flashes them, which looks cool when it's running. And then you can see which of those little pores, what they're doing, depends on the color there. But in any case, what you get when you do all these little bits that we're sequencing is we get these chunks of DNA, and at one base, that's the SNP, we get a readout of what that SNP is. So on the right there, you can see all those letters jumbled together with that one red G. So for SNP number 1, we'll call it that, that's the position we want to look at. So we want to see, is it G or is it C? Or it could be a different letter. But for each SNP, we actually get 2 answers, because remember I said we get 1 chromosome from our mother and 1 from our father. And so we actually get 2 answers. We get what that SNP is that we got from our mother and what the SNP is we got from our father. And we don't necessarily know which one it is until we go through the analysis, but I'll show you how we do that in a slide or two from now. And so this is just showing 4 individual killer whales, and you can see all the surrounding DNA there is the same in them, except where I've outlined in purple. And so on Blackberry is AA at that SNP, and Alki is TT, Mobius TA, and Suchi is AA. And so we collect these up for 42 different sites is what we're working with right now. So 42 different spots in the DNA we're looking at and seeing what are the combinations of letters. And so we can actually use this to identify the poop. So here we have some mystery poop, and we have 6, just here for ease here, we have 6 different sites, 1, 2, 3, 4, 5, and 6. And do you think you can match those up? Maybe in the chat here, we can see if anybody can identify the poop there. I'll just get the chat on here. So if we look through, we can see everything matches up with J38 cookie. And so that's who the poop is from. And here, we can also use it to determine paternity. So this is a little bit trickier, but I'll show you how we do it. Again, killer whales stick with their mothers, so the maternity is known. So that's something that can help us. Even though the paternity, so who the father is a mystery. And these DNA SNPs can tell us who the father is. So here we have J22 Oreo, and we have a new calf that hasn't been named yet. And we can see that, you know, their genotype or their letters at each of those sites is different from each other and sometimes the same, but we can figure out what we got from the mother here. So from the mother, we know that at site 1, since Oreo is GG, the calf must have gotten G from the mother. So that's a little hint that can tell us something about the father, because we know the father must have given a C. At site 2 here, we actually don't really know, because the calf is AT, but the mother is AT, so the mother could have given either of those. Here though at site 3, we know that the mother must have given a G, because that's all she has at that site, and the calf is GC. And then for the next 3, they're identical, so we know she gave one of those, but we don't actually know anything other than that. So now we can go through and look at the father, and we know that the father at site 1 must have given a C, because that calf is GC. So it's kind of fun. We can do this as, you know, a little activity and see what the father must have given. And same at site 3. GC for the calf, the mother's just GG, so we know the father is something C. We don't know what the other letter is, but the father must have had 1 C at that site. And again, for 4, 5, and 6, we can fill in the rest of what we know. So here, we can go through and see if we can guess the father. This is all we're going on. This is all we know is that the father is something C. We don't know number 2, 3, 4, and then we can go through. So right away here, is there somebody we can eliminate as a father? So I'm thinking we can probably eliminate father 2 because father 2 is just GG. So father 1 and father 3, they both have a C there that they could have given. So that keeps them in the running. Oops. Site 2, we don't actually know. It could be either. Site 3, they're the same, so that doesn't tell us much. Site 4, they both can give an A. Site 5, they both can give a T. But as we get to site 6, we know that the father gave a T, but father 1 can't. And so this can tell us, even just with that little bit of information from 6 sites, sometimes that's enough to actually get an idea of paternity. And by doing 42 of these, and we also do microsatellites, which are it's just another type of variation that we see. We measure those as well, and by putting all these different pieces together where there are differences and matching them up, we can see who the father is. And so just in summary, there's a lot that DNA can do to help us with conservation efforts. So we can bring different types of study together where we have observational data, things like the stress hormone data, and just bring it all together by kind of being that link that identifies everything. We can identify mating patterns, which is important because in different populations, it looks like, in the southern residents, that there is this propensity for just a couple of males to be doing a lot of mating. And our early analysis in the bigs or transient populations, it's a little early to tell, but it looks like that may be the case as well. So stay tuned, but we should have a bit more on that soon. And DNA can help us in other ways. So it can aid our observation efforts. So, sometimes we have somebody out in the field that sees a mating event happen, and we can look at the paternity later on and say, okay, well that is when the female got pregnant, and we can get information on that, where it occurred, what time of the year, how healthy they were at those times. So there's a lot of information that we can kind of pull together with that. And there are also things that DNA can tell us that we can't really get from other ways. So the study of killer whales didn't really exist until later in the 20th century, and but we can look at the DNA from ancient samples. So we actually have some skulls that we can extract DNA from, and things that have just been collected over the years, and even the DNA samples that we have now. Look and see how diverse are these killer whales at different sites. We can estimate the previous population size. We can estimate how isolated different populations have been from each other, and get a whole bunch of information just by getting that readout of those letters of the DNA code. And so, yeah, just want to thank funding sources. So the work that we do is funded by a little bit of Fisheries and Oceans Canada. Doug Horswell has been a great help to the Conservation Genetics Lab and the Wild Killer Whale Adoption Program, and Oceanwise. So that's my presentation there, but I'll let Danica go, but I guess we'll move on to questions.
Awesome. Well, thank you so much. I definitely learned a lot. I'm also feeling really good because it seemed like my high school biology and onward background kind of helped out with that. So I know we have a few folks who are still in elementary school in the audience, but hopefully that gave them stuff to look forward to. It's giving them a head start into all that great DNA work. And we have lots of questions, particularly lots of questions about working with poop.
Okay.
So one of the first questions that came up was, what kind of DNA extraction do you use on the poop?
Right. So that's a very good question. So there are a bunch of different methods that you can use to get DNA from anything. So they're sort of really basic, just chemical type of procedures that kind of get all the DNA. Is when we're going to figure out who the killer whale is from that poop. there's a lot of DNA in there from not just the killer whale. So there's bacteria that's been in the gut, which is actually most of the DNA that's in that poop. There's residual DNA from fish that it ate, so that gets through into the poop. And there is DNA from the skin, skin cells that are kind of sloughed off or kind of shedded, I guess, into the poop, and that's from the killer whale itself. So, um, without getting too technical, there are these tubes that you can get where you basically get all the DNA out, but then you run it through this tube, and it has a special membrane that only lets DNA from eukaryotes, or so basically like a killer whale or a mammal, like a person. If I touch the tube, my DNA would get caught in there. The DNA from bacteria just kind of flows through it, and then you kind of wash it with another chemical a little bit that just gets rid of any salt and other things, and then you just get your pure killer whale DNA, or hopefully close to it. So it's kind of a special tube that you run it through with a membrane.
Very nice. Well, lots of cool things to learn about working with whale feces.
Yeah.
Another question we had was, is the panel that you run genome wide, and how many SNPs do you identify from the feces?
So yeah, so the panel is genome wide. So there was the paper where they did the paternity in the southern resident killer whale populations. That panel is, I believe, 64 different SNPs. And I'm doing 42 right now just because that's how much money I had to buy the different reagents I needed to test those. And it's actually enough for the work that we're doing. But they are scattered throughout the genome.
Wonderful. And then we also had a few questions about, as you were identifying kind of who the father might be, for the whales out in the wild and when you're doing some of the drone work, how do you actually identify a male versus female killer whale?
Right. So the when they're younger, it's actually really difficult. So sometimes you'll see there's a new calf, and it might not be known for a couple of years or even longer than that, depending on how much they've been observed. If you can see the underside, so if they happen to breach and come out of the water, that helps because you can actually see physical differences there. But there's also, as they get older, differences in the dorsal fin. So the male dorsal fin gets really tall and straight, but the females just doesn't do that as much. So it's early on, it is tricky to tell, but as they get older, it becomes more apparent.
And as we're going to see a few more questions, I'm sure, coming in on the chat, I have one question of my own. I love that you have killer whales in the background, a killer whale on your kind of lab bench when you were showing us the sequencing equipment. So kind of where did your passion for killer whales come from, or when did it start?
So full disclosure, normally this is where the cereal for us and our kids is, but I thought I'd clean it up a little bit for the for the chat. Um, but yeah, so I guess ever since I was a kid, I've been really interested in just nature around BC. I grew up in the Okanagan, and then moved to Vancouver, and I've just really enjoyed the outdoors. But I've always been fascinated with killer whales and just sort of the ocean. And it's not something that I actually worked on in my career until recently. So most of the work I've done in terms of genetics and working with DNA is actually with these tiny little worms called C. elegans. But by working with those, they're sort of one of the main organisms or species that we use to do a lot of genetic or genomic work. And so I kind of learned a lot of the DNA side of things from that. And then at some point, after I did my PhD and did a postdoctoral work. So that's sort of like, it's not an internship. You're a scientist working in a lab, but you're kind of working on a project for a number of years. And that was again with these worms. But at some point, you kind of want to transition more into sort of a longer term career. And I thought working with killer whales sounds really cool and interesting and exciting. So when I got the opportunity to work here, I did.
Couple more questions just came in. This next one is, can you get an estimate of a killer whale's age from the photos from the drones?
So for that, hmm, this isn't really my area of expertise, but it sort of depends. It depends a little bit on which population they're from. The transients or bigs just tend to be bigger than the residents, anyways. But you can tell to some degree. But I think it would be, you could say that this killer whale is probably, um, you know, 8 or 9, but plus or minus 4 years or something. It sort of depends unless you know the history of that killer whale. But the good thing is, there's been so much there's been so much research over the last number of decades tracking these populations, not just with the more recent drone photogrammetry, but all just even pictures. A lot of these individuals we've known about. There aren't too many northern residents, definitely no southern residents that we just don't know about. So we see them pretty early on, and we can track them, and so it's less of an issue kind of identifying how old one is, because we probably already know when it was born even, and a lot of data on that.
Wonderful. And then one question came up too that you mentioned that there was an unnamed calf. How do these orcas or killer whales get their names?
So yeah, so that's a good question. It's different for each population actually. So the southern resident population, I believe it's decided by the Center for Whale Research in the United States, and I'm not sure exactly how they determine for each one what it is, what the name will be. Because it does seem more random. The northern resident population it's based on where they were observed first. So if there's a cove nearby or a point, they're generally named after that. And then the transient or Biggs population, there are just a lot more of them. They're not observed as much. They're named on a much more scientific way. So if say the mother's name is T47, her first calf is called T47A, and then T47B. And then if T47A is a female and has a calf, hers is T47A1. So it's a little less fun, but accurate.
Nice. And one question that came in is, do you know if killer whales can have twins? And if they could, would they have the same DNA?
So to my knowledge, I don't know that there have been any observations of twins in killer whales. Their DNA would not be the same unless they were from uh, the same egg. It's sort of same with humans. You can be like a identical twin, and you're going to be closer. But even if you're an identical twin, you still have some differences. So when the DNA is being replicated, so because we have so many cells, we start from just 2 cells, and then we become billions of cells. Um, Basically, the protein that copies your DNA makes mistakes, and that's how these SNPs come around in the first place. And so everybody, even if you start with the identical DNA, it's not identical pretty quickly.
Oh, it's great to see some more questions coming in. And I know we're almost, maybe about 5 minutes from wrapping up, so we'll hopefully get to answer lots. So one that just came in is, have any major differences in DNA patterns been identified between the southern resident killer whales and the northern resident killer whales?
Right, so that's a good question. The southern resident population, I've been using that SNP panel. That's what it's called, like sort of the collection of SNPs to look at in the northern residents. Um, they're going to have different sites that are kind of variable. What I found is most of those sites that we see in the southern residents that are variable are also variable in the northern residents. And what's kind of cool is because we're making sort of a big block of DNA that we're then sequencing, and our SNP is just in the middle, we also get information on the surrounding DNA, and I found a couple of these SNPs that we're looking at. And if the SNP is, say, at position 100, when I'm going through and looking at that site, I'm also seeing, at site 53, consistently there's variability in the northern resonance. So it's like an additional SNP that we're getting out of it. But you don't see that in the southern resonance at that particular site. So between the Biggs population, the northern residents, and the southern residents, at the SNP sites that we're looking at least northern and southern residents, a lot of them seem to be shared. They do have variability at the same ones. The Biggs, I'm still at the data that I was collecting yesterday will give me a bit more information on that. But there's definitely still some shared sites that have variability, but I suspect they're going to be lower. There are going to be some sites that just aren't variable in the Biggs, I think. But they'll have their own different sites that are variable that the residents may not have. But again, too early to say for sure, but yeah, we'll see.
Fair enough. Always lots more to learn. That's the great thing about this. So I think you mentioned this a little bit in one of your previous answers about identifying eukaryotic cells. But one of the questions was, do you find sequences from other organisms in the killer whale samples? And kind of how do you sort through those?
So for sure, there definitely is. One thing is the sequencing that I'm doing right now is I'm doing that PCR stuff where I'm just making lots of copies of the specific DNA that I want to look at. So when you do that, you really dilute out any other DNA that's in the sample. And there's also another step that also kind of gets rid of the starting DNA. So I'm not actually seeing any other DNA in my sequencing that's from, bacteria, plants, or fish. But if I just took the whole DNA from a poop sample, say, and ran it through the sequencer, I would get output on everything that's in there. And I may try that and just see as a test. Because of the coronavirus, I have some expiring consumables that I need to use up. So I may need to do something like that just to get a little bit of information quickly before they go bad.
A little bit of time pressure there. That's fair. And so one question just came in from a little bit earlier in the presentation as well, is kind of what do we know about paternity and how orcas choose their mates in the Biggs population?
Well, that is something that you'd have to ask Lance Barrett Leonard, he's the director of our program, but because he has a lot of observational data on that and the other populations. And it's super interesting. I'll leave it as a teaser. And maybe if he's back on giving another one of these, he can talk about that because it is very interesting. But I don't know enough about it that I can really say.
Sounds good. And we did have a great presentation from Lance a few weeks ago that we're hoping to have the recording accessible online at some point too. So there might be some answers in there.
Oh, great.
Yeah. We're almost at the end of our program. I'll give our audience another minute if they have any burning questions to put in the chat or in the Q&A. Those were some great questions. I really appreciated all of those. And you can definitely continue to learn about our killer whale research programs, whether it's at the OceanWise research page where you can get directed there by going to ocean.org. Dr. Adam Warner also mentioned the killer whale adoption program, which is a great way to learn a little bit more about the populations of killer whales that we have. So lots of great resources to check out and continue.
Yeah, and I'd really like to just say thanks to anybody that does get involved with the killer whale adoption program because it really helps, and also to Doug Horswell, who's been just a great help for the lab and for the work we do too. Just been, yeah, we wouldn't be able to do it without him either.
Wonderful. So with that, we are going to start concluding our program today. That again, thank you to our audience for so many great questions, and thank you so much, Adam, for a wonderful presentation and sharing so much of the work you do. And I am blown away that you can do the DNA sequencing at home. That was such a great insight. Definitely takes working from home to a whole new level.
That's true, yeah.
All right. So just to wrap up today, again, a big thank you to everyone, and we'd love to hear your thank yous in the chat. I'm just going to go back to sharing my screen here to tell you a little bit about what is coming up next in our programs. So from today, if you do continue to have questions or comments for us, you can find more information at education.ocean.org or visit our frequently updated page at ocean.org/learn-online. And that is where we share all of our weekly live streams, particularly our Thursday 1 p.m. Tales from the Deep, insights from OceanWise research, where you can connect to more experts like Adam today. And we will be continuing to host various programs, and we really look forward to you joining us. We'd love to see your support, whether it is online or telling your friends and family about how much you enjoy these programs. You can also visit www.vanaqua.com/saveVA to find out about more ways that you could connect with us and support us. We have had such a wonderful outpouring from the community in supporting the Vancouver Aquarium and OceanWise at this time, whether it's buying seafood chowders from local restaurants or lots of other ways to connect and support us. So thank you so much for doing so. Our next program coming up on Thursday, June 11th, is going to be Healthy Oceans Begin at Home, a community conservation toolbox with Dr. Aroha Miller. And we're really excited to be featuring a bunch of different programs next week, as Monday is World Oceans Day, and it's also OceanWise's 3rd birthday, so lots to celebrate coming up. And we're going to be celebrating Oceans Week all week long with various resources and programs, and so we hope to see lots more from you. The last thing that I would really like to thank our audience for is that at this time, we are really taking some moments to reflect on how to be more inclusive and recognizing the importance of our Black voices in our organization, in our community, and we really want to share how important it is that we are making space for that. So we have removed all of our social media at this time. We still would love to connect with you personally if you have any questions, but really we want to say how much we are open to our community, to the community, to be taking this time to reflect and think about how we can be more inclusive and making this a safe space for everyone's voices to be heard. And that as educators and lifelong learners, we are really reflecting on how we work towards allyship, towards racial justice, and more equitable social society and situations. And so we want to share learning opportunities with you. So there's a few resources here that you can go looking for to hopefully learn more, because as educators, we really want to make sure that everyone has access to those resources to be continuing to ask questions and learn throughout this whole process. So thank you so much for spending time with us this afternoon, and we hope to see you again at our next program. So, big wave to everybody. Thank you, and we'll see you soon.